Good day, this is Professor Wills, CBMD PhD, and today's Sunday, April 10th, 2022. It is 3:12 and 55 seconds, a.m. Eastern Standard Time. Today's research, autoimmune disease. So let's hop to it here. An immune fingerprint reveals path for better treatment of autoimmune diseases. Date April 7th, 2022. Source, Garvin Institute of Medical Research. Most autoimmune diseases are easy to diagnose, but hard to treat. A new article proposes using your unique immune cell fingerprint to rapidly identify which treatments will work for your autoimmune disease. Researchers analyzed the genomic profile of over 1 million cells from 1,000 people to identify a fingerprint linking genetic markers to diseases such as multiple sclerosis, rheumatoid arthritis, lupus, type 1 diabetes, spondylitis, inflammatory bowel disease, and Crohn's disease. Researchers were able to do this using single-cell sequencing, a new technology that allows researchers to detect subtle changes in individual cells. The discovery could help individuals find tailored treatments that work for them and guide the development of new drugs. The study helps us understand why some treatments work well in some patients, but not in others. It is the largest study to date to link disease-causing genes to specific types of immune cells. A trial is now underway in Sydney with Crohn's disease patients to predict which treatments will work for specific patients. Some autoimmune diseases can be notoriously difficult to treat because of our immune system's complexity and how vastly it varies between individuals. They don't currently have a good understanding of why a treatment works well in some people, but not in others. The data also provides a new avenue for narrowing down potential drug targets. The potential health and economic impacts of this research are enormous. Most rare genetic diseases are like a major car accident in the body. They are generally easy to identify and locate where they occur in the genome. But immune diseases are often more like traffic congestion, where genetic changes that hold up traffic are harder to specifically pinpoint, and this study has helped us identify the trouble spots. The greatest insight from this work will be identification of therapeutic targets and defining subpopulations of immune disease, which can then refine clinical trials to assess drug effectiveness. Our body's immune systems are designed to fight external threats, but autoimmune diseases occur when our immune systems take aim at our own healthy cells. They affect 1 in 12 Australians, are incurable, and require lifelong treatments to minimize the damage. Often patients will trial many different treatments before finding one that works for them. And some medications may be very effective in only 15% of patients, so are not recommended as a first-line treatment. And now we have a way to link treatment response back to an individual's immune genetics and to potentially screen for that 15% of patients before a clinician even administers a treatment. The researchers say their data can lower the risk associated with developing new treatments. The pharmaceutical companies may have hundreds of targets and have to make decisions about which they will take forward to phase 1 clinical trials. Knowing that 90% of potential drug candidates fail during clinical development, understanding which cell types are relevant for a particular disease is key for developing new drugs. The study provides unique insights by looking at genes in individual immune cells on an unprecedented scale. It analyzed the genomics of more than 1 million individual immune cells from around 1,000 healthy individuals, exploring 14 different types of immune cells in total. And this individual approach paints a far clearer picture from previous studies, which analyzed combined cells in a blood sample. The problems with bulk RNA analysis is that they observe an average signal, but there is vast variation in cell functions and cell types that allow the body to defend against attack. Average analysis doesn't reflect what happens in a full variety of immune cells. Researchers are working on a study of Crohn's disease in collaboration with St. George Hospital that will determine how a patient's immune genotype affects their response to different treatments, and are looking to establish new trials in a range of autoimmune diseases. It is a significant milestone of Carvin's pioneering 1K1K study aimed at showing how genetics contribute to the risk of immune disease at a cellular level. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.